This is a new video by Abron Exports for microprocessor phmeter ATC model, which is a press button touch model. Today we will discuss about the problems and how to solve them in case you accidentally do them. This first is, if when you switch it on like this, then there are two electrodes. One is a thermometer probe, another is a combined electrode that must be attached. If any one of them is not working, let us say if it is not being attached, the readings may be wrong. And if the temperature is not attached, you can see this readings may be wrong. So obviously temperature and these readings will change. So this mode should be attached. Next problem is that this electrode is dipped from one solution or any other solution directly to the testing solution. No, it should always be washed while running water by the wash bottle, and then it should be wiped. The tip should be wiped and it should be dipped about 2-3 times slowly, then in water, and rinse it very carefully. This is a tip which is not to break. This can break, so it should not touch the glass part. So very carefully, and when you see this is the second problem. Third problem is the solution is not visible in the electrode cell tube. The reason may be that this may be broken. If you put the new solution, it goes out. That means the tip is broken. Then you have to use a new tip, a new combined electrode. If it is completely filled and the water is still there, then only it will work. So it should be carefully put in the place or in the stand so that you see this is not touching the bottom. It's not touching the bottom. It should never be touched the bottom or side. And when there is a testing is going on, you should stir it by plastic or any other very slowly so that the readings are stable. Next problem is, if you have to calibrate the temperature, in the manual you are telling how to calibrate the temperature. For calibration temperature, you should press calibration and this button and then calibration. If you have to do this, read this and switch off and on, then only it goes to the calibration mode, right? And for this, press save mode. If you have done it, save mode, it is automatically converted to the selection mode. Next problem is how to go for the 3-point and 5-point calibration. So for that, switch off the power first like this, and while pressing this center mode and the save button, this is the save button, switch on it both together. Then this set calibration mode, it goes to calibration mode. And if you press cal, it goes to 2-point calibration. If you press read, it goes to 3-point calibration. That's the maximum you need. There are 5 modes by pressing save, but that is difficult, very difficult. Initially, that is required by research. So mostly we are suggesting you to go with the read mode. Read mode means 3-point calibration. And if you have done it, then immediately switch it off because you are not to calibrate it. Next point is, if you want to calibrate only, then keep both the solutions ready. Make them in 100 ml of water, distilled water, and the solution, one capsule is provided like this. This capsule should be dissolved and it should be then waited for about 2 hours. Make 3 solutions, pH 4, pH 7, and then when you switch on, connecting this, press calibration mode. Then only it goes to calibration. But be careful, it is written cal should be used to only when you need to calibrate. If by mistake press it, enter it means enter this or press it, then switch off the power immediately. Like if I press it by chance, it says 1, 2, 3 calibration start, stop immediately. Then only it comes back to the original position. Otherwise it will enter the calibration mode and then you will have to calibrate, and then it should be. Next point is, for calibration of the electrode or instrument, these all should be ready with you. Water, 3 solutions and electrode. Then you enter this, and then it says enter calibration mode. Press it for 3 seconds. 1, 2, 3. Calibration mode start. I am switching off because I am not trying to enter. There is another video by us. Go to check it how to calibrate it. Next point is, this is the calibration procedures that has been written. You enter that mode, then put it in 4 mode, pH 4, when it says enter pH 4. Then pH 7, it says enter that. pH 9, it enters. Then only calibration is done successfully. Then you can start. Once you do accidentally calibration and kept in a water, the solution will be taken as 4 pH, and then it will become a minimum 4 pH. So better avoid calibration unless it is very much required. Then there is a manual and automatic temperature mode. If you accidentally push it and go to select the manual mode, then the problem will be there. Next is, after every testing, every reading, let us say you are entering this mode microprocessor and the reading now it says 7.3. You should save it. Save means once you have saved this reading, it's sample number 6. Now if you want to read it, press it again. Now you see the sample number. It will be shown 1 sample, 2nd sample, 3rd sample, 4th sample, 5th, and these also, and 100 samples can be. If you want to clear this, switch off the power supply while pressing same mode, this mode, same mode, switch on the power supply. Don't release the save button and press read button along with it. Then this also. Then calibration button to confirm the clear. This, after this, then this. Then it will clear memory. Better avoid it unless it is very much required. Follow the manual and the instructions. It is very easy. Once again, thank you very much using the instrument, and please visit the website and also abron01@gmail.com, and this website you can send us an abronexports.com and see our other videos also. Thank you very much.